should understand the parts of the flower, the two fertile whorls, the stamens, which contain microsporangia, the carpels, which enclose the ovules, wherein there's the megasporangia that go on to germinate, where the spores germinate to become female gametophytes. So this is protection of the ovules from the abiotic and the biotic environment, as well as selectivity imposed on the pollen that can actually reach the ovules here. And we have elaborate types of pollination mechanisms or pollination syndromes in flowering plants where animal pollination is ancestral, unlike in the conifers we looked at, our main group of gymnosperms, where wind pollination occurs. And so you should study those different pollination syndromes involving different kinds of animals and wind as regards those. Those are really important in flowering plant biology. And remember that biotic pollination involves usually both an attractant, either some visual or olfactory attractant, or some kind of reward. Reward sometimes absent, but the promise of a reward is there at least, even if there's deceit pollination. And then also in angiosperms we have reduction of the gametophytes even further. The male gametophytes highly reduced, even compared to the pollen of gymnosperms. And the female gametophytes drastically reduced down to a 7-celled 8-nucleate stage. And we have then double fertilization that occurs with the pollen tubes growing down to the ovule, entering the female gametophyte, one sperm fusing with the zygote, or the egg to become the zygote, and then the other fusing with the two polar nuclei to become the endosperm mother cell that goes on to undergo a lot of mitotic divisions to become the nutritive tissue for the embryo. So the investment in the nutritive tissue happening after fertilization in this case, and the consequence of that, in part being that the nutritive tissue has a completely different basis inside a flowering plant seed than within a gymnosperm seed. This is a novel tissue, the triploid endosperm, free end endosperm. The female gametophyte's gone at this point. In the gymnosperm, though, the nutritive tissue is the female gametophyte, what's left of it, which is haploid, and was invested in regardless of whether fertilization occurred. And here we have, in both cases, the seed coat is diploid tissue from the parent sporophyte that produced the ovule initially, and the embryo, of course, is the next generation of sporophyte. Talked about a number of different mechanisms to prevent or limit self-fertilization or selfing, and one was self-incompatibility, genetic self-incompatibility. That doesn't really prevent self-pollination, but it does prevent self-fertilization of the ovules, whereas all these other mechanisms I talked about prevent self-pollination, prevent pollen from the same flower reaching the stigma of that flower. And one of these was, for example, monoece versus diece, or monoece and diece, where we have flowers of only one expression, either staminate or pistillate, but not both. That is, an individual flower is either staminate or pistillate, but in monoece we have both types of flowers on the same plant. In diece we have the individual types of flowers on different plants, like in a willow here. Also talked about spatial separation within a flower of the stamens and the stigma, and differential timing of maturation of the stigma and release of the pollen from the anthers. So those are all mechanisms that help to prevent flowers from selfing, which, remember, has some potentially negative consequences. I mean, there are plants that are self-compatible, and that has to do with natural selection that's happened at some time in the evolutionary history of those plants to allow for selfing under conditions when outcrossing wouldn't have been possible, if pollinators are limiting, mates are limiting. But in general, most plants, if they're not strictly outcrossing, they have some kind of mixed mating system where they allow for selfing under dire conditions, but they also outcross and preferentially outcross. So it's a bed hedging strategy. And so we see these kinds of mechanisms that tend to prevent at least early selfing. All right, and then talk about fruit as another major innovation of angiosperms. Remember, we talked about fruiting bodies in mushrooms, but that's really loose parlance. That's not technically a fruit is only found in flowering plants. And fruit, at minimum, represents a mature, ripened ovary, which earlier provided protection to the developing ovules, the ovules inside. But fruit does provide protection early in its development, but ultimately it's promoting dispersal of the seeds in various ways. We talked about how fruits can be dry, they can be fleshy, they can open up at maturity to release the seeds, sometimes explosively. I didn't really talk about that, but there are a lot of different mechanisms for getting the seeds away from the parent plant. And they can be fleshy as well. We had this simple categorization of fruits as either simple if they derive from one ovary of a single flower, or if the dispersal unit represents multiple free ovaries of a single flower, it's an aggregate fruit. Talk about multiple fruits where individual flowers, each of these little units of a pineapple representing an individual flower of a highly condensed flowering stalk and inflorescence. All right, also as regards pollination, so yeah, I already mentioned to review the pollination syndromes. This is just a reminder to make sure I did that as regards the attractants and rewards and morphology of the flowers. And then we got into plant structure and function on it for the rest of the semester. And so you should definitely recognize the different parts of the shoot system. The root system basically is just the roots, so the main taproot and the lateral branches of that root, and the root's lateral roots can branch as well. We have branching of branches. In the case of the shoot, of course, we have the stem. We have the leaves diverging at nodes. We have internodes between consecutive leaves. The leaves have blades where most of the photosynthesis occurs in that expanded surface, so it can capture light, and we'll do a lot of gas exchange. A stalk called a petiole. We have axillary buds as well as the terminal or apical bud. These axillary buds being stimulated at some point in the development of the plant, in many cases, to form side shoots. And we also have branching of the roots. So in the case of branching of the shoot, these buds are laid out in the apical meristem, and we have branching in that way. From the roots, we also have branching, but there are no buds. Branching occurs from that vascular cylinder, from the pericycle, the outermost ring of the vascular cylinder, and pushes its way through the cortex out into the soil through the cortex and epidermis of the primary root. So branching occurs from the vascular core in the case of roots, and in particular from that outermost tissue of the vascular core, the pericycle. We talked about modified stems and leaves. So these are such ubiquitous structures in many cases. Really, it's important to study the different types of modified stems and modified leaves. I mentioned a few modified roots, but storage roots basically and prop roots are all we talked about, although there are a lot of other modifications of roots. But things like rhizomes, these horizontal underground stems, really important for storage and asexual reproduction. Stolons above ground, also important in asexual reproduction. Tubers, the enlarged tips of rhizomes, really great storage structures within rhizomes. And bulbs, these erect underground storage structures. You should be familiar with the anatomy of the primary organs, I should say of the main three organs of the plant, the leaf, stem, and roots. We looked at cross-sections of these organs. They all have dermal, ground, and vascular tissues. You know those three types of tissues. And the cell types that we talked about, parenchyma, parenchyma, sclerenchyma, and the ground tissue, tracheids and vessels as part of the xylem, sieve tube elements, and companion cells in the phloem. But the organization of this tissue and these cell types differs between the three different organs. For example, in the stems, remember the eudicots. This is the one case where we actually compared monocots and eudicots as the stems. We didn't do that for leaves or roots, even though there are differences. Well, we did talk about in leaves how there's parallel venation in monocots and reticular net-like venation in eudicots. But the stems having pith in the center, cortex in the periphery, and eudicots with this ring of vascular bundles, phloem to the outside, xylem to the inside. But not true in the case of the monocots where the bundles are scattered throughout the stem. And we talked about meristems. You should remember the meristems of plants. Remember for primary growth that occurs from apical meristems, located at the tips of the roots and the shoots. The shoot apical meristem producing stem and leaves, and the root apical meristem producing allowing for elongating growth in both cases of the roots and the shoots. Remember, these are embryonic cells, the meristematic cells that allow for growth, indeterminate growth of plants in their vegetative tissues. Remember, the flowers and other reproductive structures like cones have determinate growth. They don't have this kind of indefinite potential for growth. And also leaves have determinate growth. They don't have meristematic cells that persist when the leaf is fully expanded. Also talked about secondary growth in woody plants, eudicots in particular. And remember, there are two types of lateral meristems that are responsible for secondary growth, which is thickening growth rather than elongating growth. A vascular cambium, which forms between the xylem and the phloem of these primary bundles and connects them in a cylinder that produces wood to the inside, which is secondary xylem, and secondary phloem to the outside. There's also the cork cambium that forms to the outside of the vascular cambium and produces periderm, mostly cork cells that are that have waxy suberin in their cell walls that makes them resistant, or makes them an impermeable barrier that replaces the epidermis that's sloughed off as the stem enlarges. So the periderm is the secondary vascular, or secondary dermal tissue. The wood and the secondary phloem, as the secondary xylem and secondary phloem, are the replacement vascular tissue during secondary growth. Remember, the wood is everything to the inside of the vascular cambium. The bark is everything to the outside of the vascular cambium. So the bark includes not only the periderm, the cork, and a little bit of parenchyma produced there, but also the phloem. Phloem's to the outside. So when you girdle a tree, when you remove all the bark, the roots starve because there's no way that the photosynthate from the leaves can move down the stem through the phloem because the phloem's been removed. Okay, down to the roots, it can't happen. We talked about absorption of water into the roots, having two different possible routes. We don't have a waxy cuticle around these root hairs that allow for absorption of water. And in fact, the soil solution can just diffuse into the cortex along the cell walls or in between in the intercellular spaces, this apoplastic route. But the only way to get across the endodermis, which is the innermost layer of the cortex, remember, is to cross the cell membrane. And that's the only way into the vascular cylinder. That Casparian strip around the endodermal cells produces a water impermeable barrier. And this prevents the vascular cylinder then from getting contaminated by soil solution. And it prevents minerals that have been accumulated in the vascular tissue from diffusing back out into the soil. We talked about water potential, and the importance of water potential as a concept for understanding how water moves within plants. Water potential is just the freedom of water to move, the potential energy of water. So the higher the water potential, the more freedom it has to move. So if we take a cell, drop it into a solution that has a higher concentration of sugars or solutes than what we have inside the protoplast, that's lower water potential to the outside. It's more bound up with solutes, and water's going to be freer to move inside the water inside the cell is freer to move, and so it moves out faster than water in the solution moves in, which is more bound up with solute molecules. Whereas we drop the same cell into pure water having higher water potential, water's going to be freer to move outside here, and it moves in faster than it moves out. But then we also have the cell wall preventing the further expansion of the cell beyond a certain level. So the pressure exerted by that cell wall counterbalances the solute potential, and we have the water potential here of 0. So the water potential has two different components, the pressure potential and the solute potential. And in the case of bulk flow in the xylem and the phloem that we mainly emphasize, remember, this is due to differences in pressure at one end of the conduit versus the other in both cases. But in this case, it's a pull from the leaves. It's a tension. The water column is being pulled up because we have more negative water potential here than we have here. Whereas here it's a push from the source to the sink. We have active loading of sugars into the sieve tube, active unloading at the other at a sink, and the water moves by osmosis from the xylem into the phloem, and we have a concentration of pressure from that movement that pushes the water down. So a push, a pull, but still bulk flow in both cases. That is movement due to a difference in pressure across the conduit. This is not movement involving crossing cell membranes. Remember, these are open channels. There's no cell membranes involved within these sieve tubes or within the xylem here. Although there is active transport in the case of the phloem with regard to loading of the sugars into the sieve tube elements. We talk about photoperiodism as one type of important biological manifestation of photoreception that has to do with flowering time onset, with many plants being either short day plants that flower when night length increases past a certain threshold here in the case of plants that flower in the late spring, early summer, or sorry, in the late summer, early fall as the days are getting shorter after the summer solstice, or long day plants that are flowering as the night length decreases past a certain threshold as we have in the late spring, early summer. And in both cases, this is due to the plant basically measuring night length through this conversion of phytochrome far red to phytochrome red gradually. So this involves the photoreceptors as the main point. Talk about hormones and how they act individually and together to either initiate various growth responses or to suppress them. And you should be familiar with these different main types of growth responses from hormones, and also know that hormones can have a variety of effects, that different plant parts have different types of sensitivities to hormones and different effects can be elicited from them, and also that hormone balance is really important in in these effects. And in particular, I talked about auxin versus cytokinin, the balance in those two types of hormones which initiate different types of responses that have to do with cell differentiation and division. So auxin resulting in elongation, cytokinin division and differentiation. That balance being important in the growth of stems and roots. I talked about ABA and gibberellins. They're balanced there too, for example, in seed germination. And finally, the last slide talked about plant defenses. Oops, darn it. This slide talking about how plants have defenses against macro herbivores with spines, cuticles, protecting the outside of the plant, secondary chemistry that can be poisonous or toxic to herbivores, actual volatile chemicals that can alert predators of your herbivores. And then I talked also about how plants can defend themselves against microbes like fungi and bacteria with a hypersensitive response and the systemic acquired resistance. That's also important to study for the final, that is. All this water transport and physiology is for the final. Okay, well, good luck tonight. And well, thanks a lot. And please let me know. Thank you. It's been a real pleasure to be your instructor for this course. And if you have further questions that come out of this, please let me know by email or come to Offshour right now. I also can meet at limited times next week. I'm gone in the middle of the week if you have questions that you'd like to meet about. All right, good luck, everybody.